Okay, Assalamualaikum. Good morning, good afternoon, everyone. So today we are going to continue our topic for this week, which is regarding the um, endocrine and nervous system. Okay, so in the previous lecture, we already learned about the endocrine system in details, right? So for today's class, we are going to study in details about the nervous system. Okay, so let me share my slide first. So endocrine system. Okay. So, endocrine system is one of the body's homeostatic control system. Okay, it controls the body homeostatic, and it contains of three parts. Okay, the nervous system are contained of three parts, which is the first one, it contains of the sensor, and the second one, it contains of the integrating centers. Okay, and the third one is the output pathway. So, in the nervous system, now if there is more interneurons in the pathway, so it will be more greater ability to integrate the information. Okay. So, in the nervous system, okay, it actually involves of the peripheral nervous system and also central nervous system. So the sensors and output pathway is actually located in the peripheral nervous system, while in the integrating centers is actually located in the central nervous system. Okay, so the sensor receptor convert the energy, okay, from the incoming stimuli. Okay, so it will receive the stimuli, okay, and then it will change in the member potentials. Okay, so here is the afferent neurons. It will conduct the signals that received by the incoming stimuli, okay, in the form of AP, action potentials, to the integrating center. Okay, so the interneurons within the integrating center, okay, for example brain ganglia, all right, it will process the informations that received by the sensor and send out the signals, okay, via the efferent neuron, okay, to the effector organs, okay, and effector organs, for example muscles or glands, okay, and it resulting in changes in behavior or physiological process. So it will go to the output, okay, as an output. Okay, so it receive the informations in the sensor, okay, travel the informations by the efferent neuron to the integrating center. Then in here they process the information and send it out, okay, through the efferent neuron to the effector organs, okay, and it will come out as a output, okay, which is in changes in behavior, any behavior or physiological process. Okay. So this is the term that we are going to use, okay, throughout this topic. The first one is the bilaterally symmetrical. Okay, so this is actually the anterior and posterior end, and also right and the left side. Also have the cephalizations. Okay, so this is the sense organ which are concentrated at the anterior end. We study about the brain. Okay, so the brain is a complex integrating center which is made up of a cluster of ganglia. Okay, so the ganglia is actually a grouping of neural cells body. Okay, nuclei is a grouping of neuronal cells body within the brain. Okay, so tract is the grouping of axon within the brain. So the nerve is an axon of efferent and efferent neuron. Okay, so that is a nerve. Okay, so this is all the term that we are going to use in this topic. So this is the review of the nerve. Okay, the structure of the nerve. So the nerve is actually composed of groups of axon from many neurons, okay, which is surrounded by successive layer of connective tissue. So this is one neuron, okay. So betul, this is one neuron. Okay, so a group of neuron, okay, we call it as a vesicle. Okay, vesicle is a bundle of neuron. So a nerve is actually composed of a group of neuron. Okay, a group of neuron, it will compose a nerve. Okay, and also it will surrounded, the nerve are surrounded by the three connective tissue, which is the first one, they have the endoneurium, and also the perineurium, and also the third one, they have the epineurium. Okay, so this is the structure of the neuron. And the mixed nerve, okay, thank you sayang. And the mixed nerve is actually contain both afferent and efferent neuron. Okay, so this is the structure of the nerve. The nervous system across the animal groups. Okay, so we are talking about the cephalizations. Okay, the specialization is occurs in most animals and becomes more apparent in more complex nervous system. So just now we already learned about the terms of cephalization. Specialization is actually the sense organ that are concentrated at the anterior end. Okay, but in the cnidarians and also echinoderms, there are exceptions. Okay, an organism with more complex nervous system have more neuron. Okay, so this is the examples of organizations of the nervous system in representative animal groups. Okay, for examples, in the cnidarians, okay, the cnidarian they have a nerve net, okay, and also nerve ring. Okay, so platyhelminthe, okay, the planarian, they have the brain and also the nerve cord. Okay, so it has some sort of a solid ventral nerve cord. Okay, this group of animal. So these cnidarians. So most nervous system are organized into three functional divisions. Okay, just now in the first slide, we already about the three functional divisions, which is the content of the sensor, the integrating sensor, and also the output pathway, right? But it not happen in the cnidarians. In the cnidarians, they have a nervous system that called as the interconnected web or the nerve net. Okay, so this is the nerve net. Okay, so the neurons are not specialized into different divisions, which is the neurons are not specialized into the sensor, integrated center or the output pathway, but they are also they have the interconnected web. Okay, which is the nerve net. So the neurons are okay. So the neuron are functionally bipolar. Is that a stimulus at any point of the organism triggers an impulse that radiates out from the stimulus. But however, even though they don't have the neurons that have the functional divisions, but it can still perform a complex behaviors. Okay, for example, in here, so this is the cnidarians. So a behavior that involves detecting a shell using a tentacles. Okay, so this is the tentacles. So this is the mouth. Okay, to grab onto the shells. Okay, so here the tentacles of the sea anemone encounter a well shell. Okay, so then the sea anemone will grab the shell with its tentacle. Okay, it will grab the shells. Then the sea anemone will release its food. Okay, we release its food from the substrate. Okay, this is the food on the substrate. So it will release the food from the substrate, and also it will perform a somersault. Point terbalik. It will perform a somersault, which is making the coordinated movement of the whole body to somersault up onto the shells. Okay, so it will attaching its food to the weld shells and releasing its tentacles. Okay, so from the tentacles from the tentacle that grabs onto the shell, it will detach back the tentacles. Okay, so thus the apparent simplicity of the cnidarian nerve must hide the substantial complexities. All right. So the vertebrate central nervous system (CNS). Okay, we are talking about the vertebrate CNS. So the in the vertebrate, they are among the most highly cephalized organism. Okay, and the vertebrate they have the unique holodorsal nervous cord. Okay, since in the invertebrate. So portion of nervous system is encased within a cartilage or a bone. Okay, as you can see from this figure. All right. And the CNS is actually comprised of the brain and also the spinal cord. While for the peripheral nervous system is actually contained of the rest of the other nervous system. Okay. Then we move to the cranial and spinal nerve. So cranials and spinal nerve form a synapse in the CNS. Okay, and in the cranial nerve is exit actually directly from the brain case. They have the 13 pairs, which is labeled with the Roman numerals, and some are efferents and some are efferents, which is some are conduct the signal in, and some are send out the signal out of the integrating centers. Okay, and the spinal nerve is emerged from the spinal cord. Okay, the cranial it exit directly from the brain case, while the spinous nerve are emerged from the spinal cord, and it actually numb based on the region of the spine where they are originate. Then we move to the gray and white matter. So the brain and spinal cord are contained of two types of tissue. The first one we have the gray matter, and the second one we have the white matter. So for the gray matter is actually a neuronal cells body, and the white matter is a bundles of axon and their myelin shell. Okay, so the spinal cord white matter is on the surface, while the gray matter is inside, which is opposite for cerebral cortex. Okay, as you can see from this figure, so this is actually a cross sections of mammalian spinal cord. So the spinal cord are contained of both gray, okay, and also white matter. So the afferents, we have the blue line is the afferents axon, okay, afferent sensory neurons enter the spinal cord, okay, which is the afferent is actually conduct the signal into the integrated center, okay, and the efferent neurons it exit the spinal cord, the red color on the ventral side, okay, which is actually it send out, okay, it send out the signal from the integrating center. Okay, so the CNS is isolated. Eh? The meninges, okay, the meninges is a layer of connective tissue that surround the brain and the spinal cord. So this is the meninges, okay. It surround the brain and also the spinal cord. The numbers of layers are vary across the taxa. For example, fish they have only one layer of the meninges, while mammal they have three layers of meninges. Okay. So within the meninges, the brain and spinal cord are float in a plasma-like fluid called the cerebrospinal fluid, okay, CSF, which is the CSF is fill the space within the meninges and act as a shock absorber. Okay, so this is very important structure, okay, in our brain, right? Because it can act as a shock absorber. And the vertebrate CNS is also physiologically separated from the rest of the body, which is within the blood-brain barrier. So the blood-brain barrier is the tight junctions in the brain capillaries. It actually function in the prevent the material from leaking out of the bloodstreams and into the CNS. Okay. So the vertebrate brain. Okay, the vertebrate brain. The brain in the vertebrate is an extension of a spinal cord. It is a hollow inside and the central cavities called the ventricle contains CSF. Okay, and they are comprised of three main regions, which is we have the hindbrain or the rhombencephalon. We also have the mesencephalon or the midbrain. Okay, so this is the midbrain, the mesencephalon, and also the forebrain is actually the procephalon. Okay, prosencephalon. Okay, so the midbrain, the hindbrain is reflex and inventory behavior. Okay, in control behavior. While for the midbrain, it actually coordinations of sensory informations. Okay, it happen in here, and it a relay center in the mammals. Okay, the midbrain. While for the forebrain, okay, the forebrain is actually the integration of the olfactory informations. So olfactory information is actually relating to the sense of smell. Okay, olfactory informations. Okay, with other senses and regulations of body temperature, reproduction, eating, and emotions. Okay, all of these process are happen in the forebrain. And in the forebrain also, they will have the process of learning and memory in mammals. Okay, so the brain size, most groups have the same major brain structure, which is they have the forebrain, they have the midbrain, also they have the hindbrain. Okay, although the structure are vary in relative of size. Okay, even though they have the same brain structure, but they are very in size. Okay, you can see from this figure. Alright, so this is the brain structure in the representative vertebrate group. For example, in the mammals, they have a very big forebrain, but very small midbrain. Okay, so this is the hindbrain. Okay, like the bony fish, they have some sort of same size of the tree structure of the brain. Okay, so this is all the brain size. Okay, even though they have the same brain structure, but they have a very in relative of size. Okay, of the brain. Okay, so this is the parts of the mammalian brain. Okay, so this is the structure, like and also the functions. Okay, the forebrain, the structure of the forebrain, the functions, the hindbrain, the midbrain, also the hindbrain. For example, the cerebrum. Okay, the cerebrum is actually functioning in information process. Okay, the olfactory bulb. Okay, it functions in the sense of smell. Okay, so we'll go one by one after this. Okay, the structure of the brain and also the functions the functions of this structure. Okay, so we go to the hindbrain first. Okay, the hindbrain is actually support the basic functions. So they have a three regions. Okay, so as you can see from here, the hindbrain, they have a three regions. Okay, so we go to the first one, the pons. The pons is actually located above the medulla. Okay, as you can see from this figure, located above the medulla, cerebrum, and also the forebrain. All right, and it controls alertness and initiates sleep and dreaming. Okay, so this is the function of the pons. Okay, in the hindbrain. So this cerebellum. The cerebellum is actually two hemispheres at the back of the brain. Okay, and this cerebellum is actually responsible for the motor coordination and also contain half of the neuron in the brain. Okay, the cerebellum. And the medulla oblongata, as you can see from here, the medulla oblongata is located at the top of the spinal cord. Top of the spinal cord. And this medulla oblongata are functioning to regulate the breathing, okay, to regulate the heart rate, diameter of the blood vessels, and also the blood pressure. So this is very important structure, right? And also the medulla oblongata are contain pathway between the spinal cord and the brain. So the midbrain, just now you're talking about the hindbrain. So this is the midbrain. The midbrain is the primary center for coordinating and initiating the behavior responses. Okay, for example, in fish and also amphibians. The size and also the functions of the midbrain are reduced in mammals. Okay, just now you see the photo, the diagram of the organism. Okay, the group of animals. Okay, they have a very difference in size of brain, right? In mammals, they have a reduced structure of the midbrain. Okay, and primarily serve as the relay center in the mammals. Okay, the midbrain, and sometimes grouped with the pons and medulla. Okay, when the midbrain are grouped with the pons and medulla, so it will terms as the brain stem. Okay, so we move to the forebrain. Okay, so the forebrain is actually function to control the complex processes. Okay, so it involved in processing and integrating the sensory informations and also involved in the process in coordinating the behavior. Okay, so in the forebrain, they have four regions. They have the cerebrum, they have the thalamus, they have the epithalamus, and also they have the hypothalamus. So the forebrain, this is why in the mammal they have a very big structure of the forebrain. Okay, because they contain of the four this region. Okay, the four very important region. So the cerebrum, we go to the first region in the forebrain, cerebrum. It's actually the outer layer. The outer layer is the cortex. Okay, the outer layer of the cerebrum is the cortex, and it's divided into two cerebral hemispheres, which is the the left side of the cerebrum. It controls the right side of the body, while the right side of the cerebrum it controls the left side of the body. Right? So it connected by the corpus callosum. So this is the corpus callosum. Okay. So this is the cerebrum, while the cortex, okay, the cortex are integrated and interprets the sensory information and also initiates a voluntary movement, okay, the controlled movement. It actually conduct in the cortex, okay, responsible of the cortex. And the cortex has taken over many of the midbrain functions in lower intervertebrates, okay, because in the lower intervertebrate they have very small midbrain. So it will the cortex will take over the functions of the midbrain in the lower vertebrate. The cortex they have a 6 layers. Okay, as you can see, they have a 6 layers here. The isocortex is the outer layer. Okay, the outer layer is actually the isocortex is necessary for cognition and higher brain functions. Okay, so in the isocortex they have more folded. Okay, in more advanced mammals. Okay, the isocortex are more folded in more advanced mammals. Okay, so they have the gyri, okay, the fold structure, okay, and also sulci, which is the groove. Okay, so this is the sulci, the groove one. So this figure is actually showing the layers of human cortex, which is the cortex in human. We have 6 layers. Okay, and the cerebral cortex is arranged in 6 distant layers, although the cellular compositions of this layer varies depending on the particular area of the cortex. Okay, the layers depends on the sites or particular layers of the cortex. Then we move to the third region in the forebrain, which is hypothalamus. So hypothalamus is located at the base of the forebrain. It maintains the homeostasis. Okay, it maintains the homeostasis in the animal and human, and the hypothalamus interacts with the autonomic nervous systems, and it regulates the secretions of the pituitary hormone. Right. So the limbic system. Okay, so the limbic system is actually a network of connected structure that lie between the cortex and the rest of the brain. Okay, so it also in the forebrain, it influences the emotions. It also influences the motivations and memory. And sometimes the limbic system we call as the emotional brain. Okay, and it includes the hypothalamus and other parts. Okay, it includes the hypothalamus, and the other part is actually the amygdala, the hippocampus. Okay, and also the olfactory bulb. Okay, so the amygdala is actually aggressions and fear responses. Okay, we respond to the fear and aggressions. Okay, hippocampus is actually function and converts the short-term memory to a long-term. All right, so this is the hippocampus, and the olfactory bulb is actually sense of the smell. So this is actually the limbic system, okay, which is influence our emotions, motivations, and also our memory. Then we move to the last regions of the forebrain, which is the thalamus. So the thalamus is actually the large grouping of grey matter above the hypothalamus, and it's actually a part of the reticular informations. Okay, and these thalamus are receive input from the limbic system. Okay, just now we're talking about the limbic system and all senses except the olfaction. Okay, so as you can see from here, so this is the thalamus. Okay, so it will receive the input from the limbic system except the olfaction. Okay, except the from here. Right. And the thalamus are release information to the cortex, and also the thalamus acts as a filter to the brain. Okay, so epithalamus is located above the thalamus. Okay, it's contained of the heavy nuclei, which is function in the communicates with the tegmentum of a midbrain. Okay, and pineal complex. Okay, epithalamus also contain of pineal complex. The pineal complex establish the circadian rhythm. Okay, and secrete the melatonin. So this is the function of epithalamus. Okay, so we have done with the structure of the brain. Okay, we have the hindbrain, midbrain, and also forebrain. Okay, so we move to the peripheral nervous system division. Okay, that just now is the CNS, so this is the PNS, the peripheral nervous system. The peripheral nervous system, it divides into the efferent branch and also efferent branch. Okay, which is the neurons that conduct the signal in and also out of the integrating center. Alright. So the efferent branch, okay, which is functioning in sending out the signal from the integrating system from the CNS. Okay, it divides into two: autonomic division and also motor division. So for the autonomic division, it divided into three category, which is sympathetic, parasympathetic, and enteric. Okay, so this is actually a major divisions of the vertebrate PNS. Okay, just now we're talking about the CNS, central nervous system. So this is actually the major divisions of the CNS. All right. As you know, that our CNS, our nervous system is divided into the two: CNS and PNS. Okay, just now in the first slide, I'm talking about CNS and PNS. Right, so this is the sensory. Okay, we go to the integrating system in the CNS, and it will send out the signal to the as an outcome. Okay, in the pathway here. Right, so it also happen in the PNS. Okay, which is the efferents. It send out the signal from the integrating center. Okay, so this is the we're talking about here, the PNS. Okay, so this is the major divisions of the vertebrate PNS. So the efferent pathway of the PNS can be further separated into two pathway: motor and also autonomic. Okay, so the motor division actually initiate the movement. Okay, initiate the movement, and the autonomic division is actually regulates the physiological functions, the outcome. Okay, outcome here. So we go to the autonomic pathway first. Okay, so it involved in the homeostasis. Okay, for example, the heart rate. Okay, to balance the heart rates, blood pressure, breathing, and also it actually the involuntary nervous system. Okay, in the previous lecture we are talking about involuntary, which is uncontrolled. Okay, we cannot control. Okay, um, the system. Okay, in the autonomic system we have three. Okay, so you still remember this one, the autonomic system, we have three. All right. So the sympathetic is most active during periods of stress or physical activities. Okay, it's some sort of fight or flight system. While parasympathetic is most active during period of rest. Okay, opposite to the sympathetic. Okay, resting and digesting system. And the third one, the third system in the autonomic pathway is the enteric. It actually dependent of other two system, right? It dependent to the parasympathetic or sympathetic system. And then in the enteric is affect the digestions by innervating the GI tracts, pancreas, and also the gallbladder. So maintaining the homeostasis. Okay, the maintaining the homeostasis is actually we're talking about the balancing of the sympathetic and parasympathetic system. So we have three features of maintaining homeostasis. Okay. Okay. So the three features is actually the first one. We have the dual innervations. The second one, we have the antagonistic actions, and the third one, we have the basal tone. Okay, for the dual innervations, it actually most internal organs receive the input from both system. Okay, from both the sympathetic and parasympathetic. The antagonistic action is actually one system stimulate while the other are inhibit. Okay, that time we go one by one. Okay, and in the basal tone, it even under resting condition, autonomic neurons produce the epi. Okay, in the basal tone, even though they are under the resting condition, but it also can produce the action potentials. Okay, so this is the dual innervation. Okay, so this is the innervation in the autonomic nervous system. Okay, so most organs are receive input from both the parasympathetic system, okay, and also sympathetic system. Okay, so this is we call as a dual innervation. Okay, antagonistic action. Okay, the antagonistic action is actually just now we're talking about the one system that stimulate while the other are inhibit, right? So the like vice versa. So for example, in here, so this is the action of sympathetic and parasympathetic nervous system in human. Okay, for example, the effector organ is the pupil of eye. So the parasympathetic, the stimulation is constrict, while the other one, the sympathetic system is dilate. Okay, they are stimulates, and the other one is inhibit. Okay, for example, the other examples. Okay, the lacrimal glands of eyes. In the parasympathetic system, they will stimulate the secretions, while in the sympathetic, it will not stimulate the secretions. Okay, so this is the effector organs. Okay, so this is the parasympathetic stimulations, and this is the sympathetic stimulations, and basically all the stimulations are vice versa. All right, so this is what we call as the antagonistic action, right? So we go to the similarities in autonomic pathway. Okay, so pathway are contained of two neurons in series, which is the preganglionic and also postganglionic. Okay, so the pathway content of the preganglionic and postganglionic. Okay, so the preganglionic it may synapse with many postganglionic neurons and intrinsic neurons, while for the postganglionics is actually the release. It will release the neurotransmitter at the effector, okay, from the varicosities. Okay, so these neurons is actually synapse with each other in the autonomic ganglia. In the autonomic ganglia. All right. So this is the structure. The figure is showing the structure of an autonomic pathway. It consists of two neuron chains, which is the preganglionic and postganglionic. Okay, just now we're talking about similarities in autonomic pathway. So now is the differences in autonomic pathway. So the differences between the sympathetic and also parasympathetic branches. So the preganglionic cells body in the sympathetic, the thoracic and the lumbar regions of the spinal cord, while the parasympathetic is actually in the hindbrain and sacral regions of the spinal cord. The ganglia locations, okay, the ganglia location in the sympathetic branches in the change that runs close to a spinal cord, while in the parasympathetic branches, the ganglia location are close to the effector. And the number of postganglionic neurons that synapse with a single preganglionic neuron, okay, in the sympathetic, the number is 10 or more, while in the parasympathetic, the number are 3 or less. Okay, the number of postganglionic neurons. Okay, we continue about the differences in autonomic pathway, the type of neurometers released, okay, at the effector. Okay, in the parasympathetic, the neurotransmitter release is the acetylcholine, while in the sympathetic nervous system, the neurotransmitter that release is the NE, norepinephrine. Okay, so this figure is showing the structure and neurotransmitter of parasympathetic and also sympathetic nervous system. Okay, so this is the differences in autonomic pathway. So only sympathetic innervations. Sorry. So although the principles of dual innervations apply to most of the target organs of the autonomic nervous system, okay, which is they will receive the information from sympathetic and parasympathetic, right? But some effector receive only sympathetic innervation. Okay, it will receive from only one, from the sympathetic innervations. For example, the adrenal medulla, it will modify the postganglionic neuron. Okay, and also in the sweat gland and also in the erector pili muscles in the skin. Okay, so the erector pili muscles is actually a tiny muscles, okay, that attach to the base of the hair follicles or kidney or most blood vessels. Okay, so they are only received from the sympathetic. So this is the sympathetic system versus the parasympathetic system. Okay, as we can see from here, the characteristic, okay, the similarities and differences between these two systems. The number of neuron in change in sympathetic, it will similar. They have two number of neuron in change, but locations of the cell body in the sympathetic, they will located in the thoracic and lumbar regions of spinal cord, while parasympathetic is actually in the hindbrain or sacral region of spinal cord. Okay, the other examples, the preganglionic neuron in the sympathetic, you will have short preganglionic, while in the long they will have long. In the parasympathetic, they will have long preganglionic neurons. Okay, like in here, the neurotransmitter released by postganglionic neuron, by post. Okay, not the by pre, by post. In the sympathetic, it will release the NE, the norepinephrine, while in the sympathetic system, it will release the acetylcholine. Okay, so this is actually the table that summarize the similarities and also differences between these two systems. Okay, so we move to the somatic motor pathway. Okay, so somatic motor pathway is actually control the skeletal muscles, usually under conscious control. Okay, the voluntary nervous system, it can control the nervous system, and some pathway are not under conscious control. For example, knee-jerk reflex. Okay, knee-jerk reflex is actually the reflex of the knee. It actually a simple reflex that causes the contractions of the quadricep muscles. Okay, the knee-jerk reflex. So the somatic pathway characteristic, it control only one type of effector, okay, skeletal muscles, and the cells body are located in the CNS. Okay, and the somatic pathway characteristic actually they are monosynaptic, which is therefore they are very long. They have only one. Okay, monosynaptic. And the axon are split into a cluster of axon terminals at the neuromuscular junctions. Okay, so this is the axon terminal, right? So in the synaptic cleft, so this is the synaptic cleft. Okay, the synaptic cleft between the motor neuron and the muscle, and then it will release the neurotransmitter we call as the ACH, the acetylcholine. And the somatic pathway characteristic, it will effect on the muscles is always excitatory. Okay. And the integrative functions of the nervous system. Okay, we talk about the reflex edge. Okay, the reflex edge is control many involuntary behavior. Okay, when we're talking about the involuntary, is actually uncontrolled. Okay, uncontrolled behavior. And the least complex integrated response, which is control the simplest types of animal behavior. Okay, it reflects. Okay, it will have rapid involuntary response to the stimuli. And also the reflex edge is actually can involve as a few as 2 neurons, okay, or more than 2 neurons, polysynaptic. Okay, so the monosynaptics, okay, so this is the monosynaptics, actually constrain only single neuron to neuron, neuron to neuron, while polysynaptics actually control synapse between more than 2 types of neuron. Okay, so this is the integrative functions of nervous system. So we're talking about the learning and memory. Okay, most animals can form memories and learn due to the plasticity of the nervous system. So the plasticity actually the ability to change both synaptic connections and functional properties of neuron in response to the stimuli. Okay, so we're talking about the learning in animals is actually the process of acquiring new information. It learned new information. While memory, we're talking about the retentions and the retrieval of the information. So when the animals are retained and also retrieval the information, and then it will become the as a memory. Okay, so the last slide is actually the invertebrate learning. For example, in the epilepsia. Okay, so the epilepsia is actually the CISLA. All right. So in the CISLA, they have for about 20,000 of neuron. Okay, so the examples of learning and memory behavior, we're talking about the habituation. Okay, so the habituation is actually the decline in response to a stimulus due to the repeated exposure. All right, so this habituation behavior, it will allow the animal to ignore the unimportant stimuli and focus on only the novel stimuli. Okay, and this will occur, the habituations will occur because of the changes in the presynaptic axon terminal. Okay, you remember? So this is the presynaptic. So this is the synaptic cleft, and this is the postsynaptic. Okay, so they occur because the changes in the presynaptic axon terminal, which is the inactivations of the calcium ion channels. Okay, then it will cause the decrease of the number of the neurotransmitter that release to the postsynaptic. Okay, so it will cause the habituations. For example, in here, so this is the head of the sisla. So this is the tail of the sisla. So this is the siphon, mantle, and also the gill. Okay, so the neural circuit governance, the gills will withdraw the reflex. Okay, so actually the sisla will exhibits a reflective withdrawal of its gills. Okay, and also its siphons in response to the weak or moderate tactile stimulation of its skin. All right. So I think that's all for today's topic. I will see you in the next week. Okay, we'll continue about the other topic of cell signaling. Okay, thank you very much. I stop recording here. Okay, assalamualaikum. See you guys.